Okay, I think this has been a very important information regarding the oncogenic role of MYD88. It's a question that cannot be addressed in human samples because human samples do have many multiple mutations, and we cannot assess specifically the role of MYD in lymphogenesis. And by introducing this single mutation into B cells, mouse B cells, we can assess the role. And the insight that we can have now after doing this transgenic mice is that MYD L265 mutation is highly prevalent in patients with Waldenström. Is not oncogenic by itself. It means it doesn't induce a transformation of primary cell by itself. It requires additional genetic mutation, although interfere with the genetic program of the B cells, but does not produce a clonal transformation of B cells.